All right, let's just jump right in. Think about this. Inside pretty much every single cell in your body, there's a complete instruction manual. It's a detailed blueprint for building and running, well, you. And of course, we call it DNA.
Okay, but here's where things get kind of wild. If you could take the DNA from just one of those tiny cells and stretch it all the way out, it would be about 2 meters long. Yeah, we're talking taller than most people, from a single cell. Just let that sink in. So that leads to a pretty big question, right? How in the world do you cram a 2-meter-long molecule into a space, the cell's nucleus, that's something like 10,000 times smaller? This is one of the most incredible information-packing problems you can possibly imagine.
Well, the answer is an absolutely brilliant packaging system. It's not just, wadded up in there like an old string. No way. It's a total masterpiece of organization at a molecular level. So let's break down how this code in the coil actually works.
So it all starts with that shape we all know and love, right? The iconic double helix. It was famously modeled by Watson and Crick, who were building on the absolutely crucial X-ray work from Rosalind Franklin and Morris Wilkins. It's basically a twisted ladder with 2 long strands that actually run in opposite directions, a feature scientists call anti-parallel.
Now, the rungs of that ladder are where the magic really happens. They're not random at all. They're made of 4 chemical bases, A, T, C, and G. And the pairing is super specific. Adenine, or A, always pairs with thymine, T, and guanine, G, always pairs with cytosine, C. They're held together by these things called hydrogen bonds. And fun fact, the GC pair is a little bit stronger because it has 3 of those bonds instead of 2. This simple rule is basically the foundation of all of genetics.
So how do you pack that 2-meter ladder? It happens in stages. First, the DNA wraps itself around these proteins called histones, kind of like thread around a spool. That forms something called a nucleosome. Then, these little nucleosome beads coil up into an even thicker fiber. After that, those fibers form big loops that get anchored to a protein scaffold. And finally, the whole thing gets coiled and condensed even more to form that classic X-shaped chromosome you probably remember from science class. It's an amazing process that achieves a 10,000-fold compaction.
Okay, so we figured out how to pack the blueprint. Awesome. But now the cell has another massive challenge. Before a cell can divide into 2, it has to make a perfect full copy of that entire 2-meter-long blueprint. How does it even begin to do that?
Well, the process is called DNA replication, and it has this really cool feature. It's semi-conservative. Now, that's a key idea. It just means that when the DNA gets copied, that double helix unwinds, and each of the original strands becomes a template for building a new partner. So every new DNA molecule you make is half old, half new. And that is just a fantastic way to make sure the original information is preserved super accurately. Now, to get this done, the cell calls in a whole team of molecular machines, these enzymes. You can think of them like a little construction crew. First up, you've got helicase. That's the unzipper. It just unwinds the 2 strands. Then comes the star of the show, DNA polymerase. This is the master builder that reads the old strand and brings in all the right new bases. But here's the catch. Polymerase can't just start from scratch. It needs a little starting block, an RNA primer, which is laid down by the initiator, primase. And finally, you've got DNA ligase, which is like the superglue that seals up any little gaps at the end.
But wait, there's a wrinkle. Remember how the 2 DNA strands are anti-parallel, running in opposite directions? That creates a bit of a problem because our master builder, DNA polymerase, can only build in one direction. So one strand, which we call the leading strand, gets copied in one long, smooth, continuous piece. Easy enough. But the other one, the lagging strand, runs the wrong way. So it has to be copied backwards in a bunch of short little chunks called Okazaki fragments. And then our friend DNA ligase comes in and stitches them all together.
And even with all this complexity, the whole thing is unbelievably accurate. Thanks to other enzymes that actually proofread the work as it's being done, the final error rate is something like 1 mistake for every billion bases copied. That's insane. It's like typing out an entire encyclopedia set 100 times and only making a single typo. Okay, that brings us to the next big problem. A cell doesn't need to read that whole 2-meter-long blueprint all at once. Most of the time, it just needs the recipe for one specific gene to build one specific protein. So how does it find and copy just one recipe from a library of thousands? The solution is a really elegant process called transcription. Instead of pulling the giant, precious master blueprint off the shelf every time, the cell makes a temporary disposable photocopy of just the gene it needs right now. This copy isn't DNA, it's a slightly different molecule called messenger RNA, or mRNA.
This is brilliant because it keeps the original DNA master copy safe and sound inside the nucleus.
But that first RNA copy? It's kind of a rough draft. Before it can leave the nucleus and actually be used, it needs some editing. In complex cells like ours, what scientists call eukaryotes, this happens in 3 main steps. First, a protective cap gets added to the front, kind of like a little helmet. Then, any non-coding sections, called introns, get snipped out. This is a process called splicing. And finally, a long tail of A bases gets added to the back, which helps it last longer and get where it needs to go.
And that splicing step I just mentioned, this is where things get really, really clever. The cell can actually splice that same gene in different ways. By mixing and matching the useful coding bits, or exons, a single gene can produce a whole bunch of different versions of a protein. Get this, from our roughly 20,000 genes, this alternative splicing lets our cells create over 90,000 different kinds of proteins. It's just an incredible example of biological efficiency.
So, we've packed the blueprint, we've copied it, and we've made a disposable messenger. Now for the grand finale, translating the language of that RNA message, which is written in chemical letters, into the physical 3D structure of a working protein. The genetic code itself has a pretty simple set of rules. The mRNA gets read in 3-letter words called codons. Each of these codons specifies one of the 20 different amino acids, which are the building blocks of all proteins. And just like any language, there's punctuation. There's a specific codon, AUG, that always means start. And there are 3 different codons that signal stop, telling the machinery that the protein is finished.
This whole process is called translation, and it happens at a molecular factory called the ribosome. The ribosome basically clamps onto the mRNA message and starts reading it, codon by codon. But how does it know which amino acid goes with which codon? That's the job of another cool molecule, the translator, called transfer RNA, or tRNA. Each tRNA carries a specific amino acid and has a 3-letter code that matches an mRNA codon. It's like the ultimate molecular matchmaker.
So, we've gone from DNA to RNA to protein. You know, it seems like a pretty straightforward assembly line, but the reality is so much more complex. This whole system is actually part of a huge, highly regulated network that's constantly controlling which genes get turned on or off and when.
For a long time, the classic view, first laid out by Francis Crick, was called the central dogma of molecular biology. It's a simple, elegant flow of information. DNA makes RNA, and RNA makes protein. And for decades, this was really the bedrock of how we understood everything. But that simple idea hides a much, much bigger story. It turns out that only about 1.5% of our entire DNA blueprint actually codes for proteins. A huge amount of it, almost 80%, gets transcribed into RNA that never makes a protein. For a long time, this stuff was called junk DNA, but we now know it's anything but. It's a massive hidden layer of control, basically the complex operating system that regulates that little 1.5% of the code.
So I think the real takeaway here is this. Our genetic blueprint isn't just a static book of recipes. It's a dynamic, interactive network that's constantly responding to the world around it. We've learned how to read it, and now we're just starting to learn how to edit it. Which leaves us with a final, really fascinating question. What parts of this incredibly intricate network will we learn to rewrite next?